Bismillahirrahmanirrahim. In the name of Allah, the most gracious and the most merciful. This is me, Namra Sarwar, presenting my presentation topic, which is vitamins, its classification, synthesis, and methods used for vitamin research. Presented by me and presented to respected Dr. Hammad Majeed. These are the contents of my presentation, which includes introduction, water soluble vitamins, fat soluble vitamins, synthesis of vitamins, which further includes the synthesis of vitamin A, D, E, and K. Then, vitamin deficiency disease, methods used in vitamin research, which further includes physiochemical methods, microbiological assay, animal assay, determination of vitamins by Hplc, top vitamin manufacturers, vitamins and dietary supplements, market historical trend. Let's begin with the introduction. Vitamins are substances that our bodies need to develop and function normally. They include vitamin A, C, D, E, and K, choline and B vitamins, which further includes thiamine, riboflavin, niacin, pantothenic acid, biotin, vitamin B6, vitamin B12, and folic acid. Vitamins are a group of organic nutrients of various nature required in small quantities for further multiple biochemical reactions for the growth, survival, and reproduction. Our body needs vitamins to function properly. We cannot produce most vitamins ourselves, at least not even in sufficient quantities to meet our needs. Therefore, they have to be obtained to the food we eat. Water soluble vitamins. Water soluble vitamins, as the name indicates, these are easily soluble in water. And it includes vitamin B1, B2, B3, B5, B6, B7, B9, B12, and C. B1 named as thiamine, B2 named as riboflavin, B3 named as niacin, B5 named as pantothenic acid, B6 named as pyridoxine, B7 named as biotin, B9 as folate, B12 as cobalamin, and C as ascorbic acid. Now the fat soluble vitamins. Fat soluble vitamins are not soluble in water and are similar to oil. They contain vitamin A, vitamin D, vitamin E, and vitamin K. The function of vitamin A is to keep the skin and mucous membrane healthy, prevent night blindness, and promotes growth. And the sources of vitamin A includes butter, dark green and yellow fruits and vegetables, egg yolk, liver, whole and fortified milk. The next one is vitamin D, which behaves as to build strong bones and teeth, and their sources are egg yolk, fortified butter, margarine and milk, and especially the sun. Vitamin A acts as an antioxidant to protect cell membranes, and the sources of vitamin E includes eggs, liver, salad oils, whole grain cereals. And the last one is vitamin K, which helps in blood clotting. And the sources of vitamin K are cauliflower, egg yolk, and organ meats. Now the synthesis of vitamins. Vitamin A synthesis. Vitamin A is present in two forms, A1 and A2 carotenoids, such as alpha, beta, and gamma carotenes, and the tryptoxanthin act as provitamin form of vitamin A. Beta carotene is the most potent provitamin form. Beta carotene is made up of 8 5-carbon isoprenoid units like to form a long chain of 40 carbon atoms with an ion 1 ring at each end. Hydrolysis of beta carotene yields two molecules of A1. Cleavage occurs at the midpoint of carotene in the polyene chain connecting the two ion 1 rings. And then the conversion of beta carotene into vitamin A takes place in liver of fishes and mammals. Vitamin A is a complex primary alcohol called retinol. The terminal hydroxyl group is mostly esterified with fatty acid. Basically, The beta carotene, which consists of two molecules of retinol linked at their aldehyde ends, and it is also referred as provitamin form of vitamin A. Here is the reaction mechanism in which the cleavage occurs in the midpoint of the beta carotene, forming two products of vitamin A1 retinol. The next one is the synthesis of vitamin D. D3 cholecalciferol is produced from 7-dehydrocholesterol 7-DHC through a two-step process in which the B ring is broken by ultraviolet light with a spectrum of 280 to 320 UVB. Radiation from the sun forming pre-D3 that isomerizes to D3 in a thermosensitive but non-catalytic process. And you can see the reaction in which the 7-dehydrocholesterol in the presence of ultraviolet light converted into pre-vitamin D3, and then the vitamin D3 cholecalciferol is further converted into the most stabilized form calcitriol, which is 125 dihydroxycholecalciferol. Here is the mechanism which determines these are the pericyclic reactions. They do not need particular high temperatures, but indeed they can be catalyzed by either the high photons or temperature. They are not reactions, rather just intramolecular rearrangements of electrons over a chain of connected atoms. The first reaction is a photolytically catalyzed electrocyclic reaction that occurs in the opening of the six-membered ring. They are very common. The second reaction is a thermally catalyzed 1,7 sigmatropic migration of the hydrogen. The rate is dependent on the nature of the nucleus that moves. Quantum tunneling will indeed enhance the rate of its non-quantum counterpart, but it is not the driving force of the step. Next is the synthesis of vitamin K. Vitamin K exists in two natural forms. First one is phyloquinone, which is also called K1, and menaquinone, which is called as K2, with 4 and 6 isoprenoid units respectively. In phyloquinone, 3 isoprenoid units are dehydrogenated. Menadione K3 without an isoprenoid chain is a synthetic derivative. The first one is the structure of phyloquinone, and the second one is the structure of menaquinone. Here is the reaction mechanism of vitamin K. Vitamin K is involved in the process of blood coagulation, being required for the synthesis of prothrombin and factors 8, 9, and 10. Vitamin K is involved in the post-translational modifications of these proteins, consisting in the gamma carboxylation of glutamate residues. Vitamin K is the coenzyme of the carboxylase enzyme and acts in its reduced hydroquinone form. Synthesis of vitamin E. The synthesis of vitamin E occurs in 4 steps. The first one is conversion. The second one is a reduction. The third, inhibition. And the fourth is ferroptosis and inflammation. These are the mechanism steps of synthesis of vitamin E. The anti-ferroptotic effect of vitamin E results from a 4-step biochemical mechanism. The first step involves the oxidative hydrolysis of alpha tocopherol to alpha tocopherolquinone. The second step involves the reduction of alpha tocopherolquinone to alpha tocopherol hydroquinone. Inhibition of 15-LO via reduction of its non-heme iron center to the inactive iron state by alpha tocopherol hydroquinone. Inhibition of the ferroptosis cascade by blocking formation of lipid peroxidation products, which includes polyunsaturated fatty acids PUFA, PUFAOOH, hydroperoxidated polyunsaturated fatty acids, and hydroxylated polyunsaturated fatty acids. The non-metabolizable isosteric analog of vitamin E retains its antioxidant activity but does not exhibit ferroptosis. Here are the vitamin deficiency diseases. Vitamin deficiency diseases includes vitamin A deficiency diseases, vitamin D deficiency diseases, vitamin E deficiency diseases, and vitamin K deficiency diseases. Vitamin A causes night blindness, dry eyes, rough skin, slower bone growth, and improper tooth development. Vitamin D causes cancer, rickets, and softening of the bones. Vitamin E causes muscular weakness and neurological dysfunction. And vitamin K causes blood clotting difficult and reduces the bone density. Next is a vitamin toxicity. Taking an excessive amount of any one vitamin can cause serious health problems. A condition generally referred to as hypervitaminosis or vitamin toxicity. Some medications can also increase the risk of vitamin toxicity either by increasing the body's absorption of a vitamin or by containing vitamin based compounds. Vitamin A toxicity commonly affects the skin, causing reddening, irritation, and patchy peeling. Excessive consumption of animal food sources like liver or fish oil, in addition to supplements high in performed vitamin A, increases the risk of vitamin A toxicity. Here are the methods used in vitamin research. And the common methods to determine vitamin content are physiochemical methods, microbiological assay, and animal assay. Physiochemical methods. The amount of vitamin in a foodstuff can be established by studying the physical or chemical characteristics of the vitamin. For example, a chemical reactive group on the vitamin molecule, fluorescence, absorption of light at a wavelength, characteristics of the vitamin, or radioisotope dilution techniques. The next and the second technique is microbiological assay is applicable only to B vitamins. The rate of growth of a species of microorganism that requires a vitamin is measured in growth media that contains various known quantities of a foodstuff preparation containing unknown amounts of the vitamin. The response to the unknown amount of vitamin is compared with that obtained from a known quantity of the pure vitamin. This method has following advantages. This method determines the potency. This method controls antimicrobial chemotherapy. Good in vitro and in vivo correlations are provided by this method. This method is accurate, inexpensive, and convenient. This method is easy to interpret results to the microorganisms. Animal assay. All of the vitamins, with the exception of vitamin B12, can be estimated by the animal assay technique. One advantage of this method is that animals respond only to biological active forms of the vitamin. On the other hand, many other interfering and complicating factors may arise, therefore, experiments must be rigidly standardized and controlled. In a growth assay, the rat, chick, dog used specifically for niacin vitamin and pig used specifically for vitamin C ascorbic acid usually are used. Determination of vitamins by Hplc method. Traditional Hplc method. This method involves the reverse phase Hplc and it is well suited technique for vitamin. Water soluble vitamins are determined using an aqueous mobile phase whereas the fat soluble vitamins are determined using organic solvent mobile phases. Commonly used buffers for the separation of water soluble vitamins are phosphate, formic acid, and acetic acid. NARP retention is commonly used for fat soluble vitamins so that the vitamins are soluble throughout the analysis. A typical NARP mobile phase consists of a polar solvent, acetonitrile, a solvent with lower polarity, for example dichloromethane, to act as a solubilizer and to control retention by adjusting the solvent strength. The second process is the reported Hplc method, and there are numerous methods for the simultaneous determination of water and fat soluble vitamins. The vitamins were separated on the thermoscientific acclaim polar advantage 2 PA2 column with a single injection using an aqueous to non-aqueous mobile phase gradient. Due to the large differences in sample preparation method, this method is insufficient in analysis of solid samples such as multivitamin tablets. In the work presented here, the retention behaviors of water and fat soluble vitamins are investigated on three types of reverse phase column. They claim PA, PA2, and C18 columns. Top 5 vitamin manufacturers across the globe. First one is BASF-SE, is a German chemical company that has a diverse portfolio. Their product for human nutrition includes dietary supplements, functional nutrition, medical nutrition, infant and maternal nutrition, beverages and foods. They are of the top vitamin manufacturers who manufactures enzymes, glycinates, carotenoids, organic acid, vitamins and clay products. The second one is DSM. Royal DSM is a Dutch multinational company that provides products and services related to health, nutrition, and materials. The third one is Lonza. It's a life sciences company and also the world's leading supplier of vitamin B3 for over 40 years. The company provides integrated solutions to buyers across markets such as pharmaceutical, biotech, and specialty ingredients. In recent times, the company has invested heavily in studies to understand the role supplemental niacin plays in both human and animal health. The fourth one is Adisseo. Adisseo is a world leader in additives and nutritional solution for animal feed with offices all around the world, such as France, Georgia, Brazil, China, and Singapore. Among the leading vitamin manufacturers for animal feed, the company sells its vitamin under the brand name Microvit. ADM. Last but not the least, Archer Daniels Midland is a global food processing and commodities trading corporation which procures, stores, processes, transports, and merchandises agricultural commodities and products. ADM is also the largest producer of natural source vitamin E and mixed tocopherols. The company also boasts the broadest vitamin E product line with alcohol, tablet, and compounding applications, and acetates and succenates in a wide range of manufacturer friendly forms of capsules. Here are the vitamins and dietary supplements market historical trend. The market trend for vitamins and dietary supplements is driven by the increasing attention to health and prevention by the customers, the greater customization of needs for different segments of the population, increased healthcare costs and search for alternatives to cure specific problems. The growth in demand for supplements is mainly driven by probiotic supplements, fatty acids, that is, fish oils, and protein supplements. Herbal or botanical supplements usage has emerged as a popular complementary and alternative medicine or supplement to modern medicine. Rising consumer awareness regarding the severity of digestive disorder stimulate the growth of the enzyme segment. These are the references from which I had taken the material for my presentation, and Jazakallah khair. Thank you so much.